Hi there, my name's Wendy, and I wanted to walk us through a quick yarn demonstration of gene insertion using gene editing. And if you want to follow along with me, let me just tip the camera here. You don't need too many supplies. I'm using yarn today, but anything that you have that's in a couple different colors that you could roll out into a string would definitely work. And so you need 3 colors of yarn and also a pair of scissors, which we will be using to get through this. And what I have here in green and gray are my 2 strands of yarn that are going to represent my DNA today. In yellow, in these 2 batches, these are going to represent my genes of interest, and I'll just focus on the green strand for now. As you can see, the gene of interest is already attached to the green strand here, because when we are creating this gene that we want to insert, we do actually add a couple guides at the end of it so that we know exactly where it's going into the DNA and so that we know exactly where it will match up. So I'll take a quick pause here for a second, or if you want to pause the video, please feel free to grab some supplies and follow along with me. Okay, so like I said, the green and the gray strands here are representing our 2 strands of DNA. And what we can do with different chemicals is that actually act a lot like scissors, is we can actually create these double strand nicks, just like that. And I knit my yarn into some cord here, and it's actually unraveling a little bit, which is actually a little similar to what our DNA does, but we won't go into that today. Anyway, once we create this double strand break, there's a little room to spread these apart, and there's a little room for these to kind of float around. And what we want to do is take advantage of this break here. Like I said, we've got our green guides on there so that we can insert our strand of interest. Just like that. And what I should have had with me as well is a bit of glue, actually, because what our cells will do is they'll actually join and attach these ends together so that it's one single strand all over again. And then the great thing about our bodies is that we're really good at repairing our DNA, and we're really good at matching our cells together. So what we'll do after this is that once this strand is inserted, our body is actually great enough to actually make the exact match needed and copy in all of those little other bits of DNA so that we once again have 2 matching strands of DNA, but now with our gene of interest inserted. So I hope you enjoyed that quick little demonstration, and it starts to get into how gene editing and personalized medicine can be really helpful in treating diseases. Thanks so much for following along.